You are welcome to my another session of Cytology class. The topic of discussion of this session is on types of chromosomes. In the previous sessions, we have studied the structure of a eukaryotic chromosome and chromosome organization, that is how the DNA has been packaged with the help of nucleosome concept. Now let us discuss on how the chromosomes have been classified. As we are aware, chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Each chromosome is made up of a protein and a single molecule of DNA, that is deoxyribonucleic acid, and is passed from parents to the offspring. DNA contains the specific instructions that make each type of living creature unique. There are various factors based on which the chromosomes have been classified. For example, based on the function. Secondly, based on the number of centromeres present. And thirdly, based on the location of centromere. And another aspect is on holocentric chromosomes. The first aspect, the classification of chromosomes based on function. Based on the function of the chromosomes, they can be divided into two categories. One is called autosomes and another one as sex chromosomes. Autosomes are present in all cells controlling somatic characters of an organism, whereas sex chromosomes are involved in the determination of sex. Genetic traits that are linked to the sex of the person are passed on through the sex chromosomes. The rest of the genetic information is present in autosomes. For example, in humans, we have 23 pairs of chromosomes in their cells, of which 22 pairs are autosomes and one pair of sex chromosomes, making a total of 46 chromosomes in each cell. Based on the number of centromeres. The first one is acentric. That means the chromosome is without any centromere. Acentric chromosome is a very rare occurrence. It may arise due to unequal breaking of chromosome arm into two so that only one part has the centromere while in the other part centromere is absent. And sometimes due to the lack of centromere, acentric chromosomes are not able to attach with the spindle fibers and hence they do not take part in cell division. And ultimately, the cell eliminates the acentric fragments. Such chromosomes represent freshly broken segments of the chromosomes which do not survive for long period. Now the second type is monocentric. Here chromosomes with only one centromere. And the third type is dicentric. That is, a chromosome has two centromeres. Dicentric chromosome may be produced as a result of translocation, paracentric inversion, etc. If the two centromeres tend to move to opposite poles during anaphase, the chromosome breaks. Rarely, a new centromere may appear on the chromosome, resulting in an abnormality. Such a centromere is called as a neocentromere. Dicentric chromosome is reported in the cells of wheat. Now, polycentric chromosomes. That means each chromosome has more than two centromeres. And these type of chromosomes are called as polycentric. And polycentric chromosomes are found in plants, for example, in Luzula purpurea, which belongs to family Juncaceae. However, in many insects, the centromere activity is non-localized and spread over the entire chromosome length. In such cases, the centromere is called as diffused centromere. Polycentric chromosomes often break into a number of smaller fragments, and each small fragment functions independently. So this is the schematic representation of chromosomes with multiple centromeres. A centrosome with two active centromeres, dicentromere or dicentric chromosome, is typically unstable. A twist between the sister chromatids within the region between both the centromeres leads to metrolytic spindle attachment of the two kinetochores on the same chromatid, resulting in anaphase bridge and subsequent chromosome breakage. And in the diagram B and C, stabilization of a dicentric chromosome can occur by inactivation of one centromere so that chromosome behaves as monocentric. In the C, when the close proximity between the two active centromeres enables them to behave as one functional unit. That is, in the D, it is metapolycentric chromosome with three functional centromeres within one constriction. So now the classification of chromosomes based on the location of the centromere. First one is telocentric, rod-like chromosome, where the centromere is present on the proximal end. No p-arm or short arm is present. Telocentric chromosomes are not found in human beings. Acrocentric, these are rod-like, and centromere is present at one end, giving rise to one very short arm and another exceptionally a long arm. Submetacentric, they are also called as L-shaped or J-shaped chromosomes with centromere near the center of the chromosome, giving rise to two unequal arms. And the last one is metacentric or V-shaped chromosomes where centromere is in the middle, giving rise to two equal arms. Holocentric chromosomes. Holocentric or holokinetic chromosomes have a diffused kinetochore, making these chromosomes able to bind to the microtubules along their entire length. They possess multiple kinetochores all along the length rather than a single centromere, a typical of other chromosomes. These chromosomes are also called as holokinetic, and this is because during cell division, chromatids move apart in parallel and do not form a classical V-shaped figure, which is a typical of monocentric chromosomes. And holocentric chromosomes are currently reported in about 800 diverse species, which includes plants, insects, nematodes, etc. Because of their diffused kinetochore, holocentric chromosomes may stabilize chromosomal fragments, favoring karyotype rearrangements. However, holocentric chromosomes may also present limitations to the crossing over, causing restrictions of the number of chiasma in bivalents and may cause a restructuring of meiotic divisions resulting in inverted meiosis. Meiotic versus mitotic segregation in an holocentric chromosomes. This diagram shows the chromosome segregation during mitosis and meiosis. During mitosis, chromosomes are holocentric with a diffuse kinetochore along the length, whereas during meiosis, microtubules are directly embedded in the chromosome ends, but the specific chromosome ends is chosen randomly. At meiosis 1, cohesion between homologs is degraded, allowing the separation of homologous chromosomes to opposite poles. During meiosis 2, the opposite end of the chromosome is used to attach to the microtubules. At the end, the transition to the anaphase, cohesion between paired sister chromatids is eliminated, allowing their segregation to opposite poles. Now lastly, about the function and significance of chromosomes. Phylogeny and taxonomy. The number of the chromosomes is constant for a particular species. Therefore, these are of great importance in the determination of the phylogeny and taxonomy of the species. And the second one is the genetic code and storage. Chromosome contains the genetic material that is required by the organism to develop and grow. DNA molecules are made of chain of units called genes. Genes are those sections of the DNA which code for specific proteins required by the cell for its proper functioning. Then sex determination. Humans have 23 pairs of chromosomes, out of which one pair is sex chromosome. Females have two X chromosomes and males have one X and one Y chromosome. Then control of cell division. Chromosomes check successful division of cells during process of mitosis. The chromosomes of the parent cells ensure that the correct information is passed on to the daughter cells required by the cell to grow and develop correctly. And lastly, the formation of proteins and their storage. The chromosomes direct the sequences of proteins formed in our body and also maintain the order of DNA. The proteins are also stored in the coiled structure of the chromosomes. These proteins bound to the DNA and help in proper packaging of the DNA. Thank you.